Well, good evening, everyone. A pleasure to welcome you here. My name is Mark Sargent. I serve as the provost of Westmont College, and on behalf of Westmont and behalf of the Westmont Foundation, we're glad to have you here for the second in our series of Westmont Downtown Lectures. I do want to mention our third in the series, which is going to be something very, very special in January. On January 9th, we are having a special Westmont Downtown at the New Vic Theater, where we will be doing a commemorative program 1 year after the debris flow. And it will be looking back in retrospective on this year. It'll be commemorative in light of some of the tragic events, and also educating ourselves about our environs, and something that marks the occasion in an appropriate way with the community members as well. At the outset of my remarks, I would like to express the deepest sympathies on behalf of Westmont College to the families and the neighboring institutions that lost students and lost loved ones in the shooting in Thousand Oaks this morning. You know, our heart always aches when anyone's life ends in violence, and when you think of the youth cut off at the beginnings of their lifetime, it's particularly traumatic. So we join them with our prayers and our sympathies, and also our just commitment to the importance of education in reducing violence in our world. And we will certainly share our sympathies with our sister institutions. My task right now is much more pleasurable. I have the honor of introducing to you Beth Horvath, who is an associate professor of biology at Westmont, and she is our speaker this evening. She's a graduate of Westmont, and she also completed her master's degree at Cal State Long Beach in marine sciences, and then returned to Westmont to teach in 1978. And for many years, she has also served as a research associate in the invertebrate labs at the Santa Barbara Museum of Natural History. She has become quite an expert on life under the sea. In fact, she consults with over 15 government or non-government agencies in the identification of sea corals. As she tramps about in the tide pools in her waders, Beth knows exactly what crevice to look in to find the marvelous sea life. In the lab, she will examine 2 specimens of coral that the average person, even some experts, judge as identical. But then she can identify the nuances that separates them as different species. The Smithsonian and the Australian government will regularly ship collected samples to Beth and ask her to identify the species. Recently, she was appointed as the main scientific expert on the National Oceanographic and Atmospheric Administration's research project that is characterizing deep sea coral and sponges off the California coast. She will be guiding shipboard crews to know how best to use the latest technology available so as to inform what to look for, how to find it, go about collecting it, recording it, and photographing it while they're under the water. Though she has such extensive credentials and experience and recognition for her work from around the world, Beth has a big heart for teaching and mentoring undergraduate students. Some former students I know are here tonight to celebrate this event with you. Some students will base their entire schedule around taking whatever class she is teaching. Students seek her advice when she is not their instructor because they value the wisdom and the ways in which she's always trying to help out. One of her colleagues, Steve Giulio, remarks, "Anyone who sees Beth Horvath in action, even for just a few minutes, will know that she is a born teacher." Apparently every year at the biology department's annual breakfast, many seniors will recall that their fondest moments at the school were times with Beth, when they have explored the intricate diversity of life in a tide pool, or raced down the sand dunes at Montana del Oro, or spent a Valentine's Day watching elephant seals mate at San Simeon. In all that she does in the lab, in the classroom, or in those waders along the ocean, she helps students discover the wonders of our world, and we're a pleasure to have her come tonight to share some of that wonder with us. So please welcome Beth Horvath. You should come and speak.
Well, Good evening, and I'm really pleased to see so many of you here. We have a really exciting thing that's happening in the next few years. NOAA is going to be undergoing a 2-year survey project along the coast of Washington, Oregon, and California. We are going to be looking at a lot of seabed that has not been looked at, about 110 miles out or more from shore, anywhere between 300 feet to many thousands of feet. There is a lot of bottom topography that has not been looked at. You would think for all the time that people have spent looking at marine life in California. It's kind of a hotbed where a lot of Mexican forms interact with a lot of forms from the Pacific Northwest, right in the middle of the state, where the state has that funky little bend to it at Point Conception. You would think that with all the years that have been spent studying this area, we'd know a lot more than we do, but there's a lot of work to do. And we are really happy that we managed to get this 2-year funding. So, my title, which was not the title that we used to advertise, it was a little long, and I didn't really think that people would really understand just exactly what is she going to talk about. So, but all of this will become very clear. There really are bubblegum gardens, there really are plastic forests, and there really are sugar daddies down at the deep. So, I just want to remind you a little bit of some of the key physical factors that organisms living at depth have to contend with. You are probably going to think that there's probably not a whole lot of light down there, and you would be right on that one. So there is very little light. There's a little bit of glow that comes from some of the molten magma that's coming up through cracks in the Earth's crust, but basically it's absolutely dark. It's hard to imagine any kind of environment where there's a total absence of light, but this is what we're dealing with. Temperature, it's pretty dang cold down there. It's actually cold enough to freeze, but for the fact that you have a body of water that's salty. So the salt lowers the freezing point, so it doesn't actually freeze, which is good for us. But the cooler water temperatures allow for a lot more oxygen to be dissolved in the water, and that's quite beneficial to the organisms that are living down there. Lots of pressure, tons and tons of pressure. You think about this massive body of water sitting on top of organisms that are a mile or two down on the ocean floor. That's a lot of water sitting on top of you, and many of you in our community, many of you and many in our community know the power of water. We experienced it in something of a negative context last January. So it is a powerful force. The bottom topography can be anything from exceedingly flat, what are called abyssal plains, with a lot of sediment accumulating on the bottom, but there's a lot of volcanic formations, there's submarine seamounts, there are very sheer-sided cliffs, there's a lot of three-dimensional topography. So all of those things have a part to play in the kinds of organisms that live there. And believe it or not, there's actually even currents of water that flow very discreetly and very intently in various parts of the ocean. We usually tend to think of the deep sea as being a very stable, non-moving body of water. We don't think of waves and tides and all of those sorts of things, which they don't experience. But there are actually a lot of currents that flow along the bottom, and a lot of animals actually depend upon those movements of water. So, things that NOAA is really concerned about with regards to some of those physical factors is the fact that, and you've probably heard this a great deal, that there is a slight increase in water temperature. It's particularly noticeable in shallow water, but unfortunately we are also starting to see changes in water temperature at great depth. And that was something that we did not expect to see for many, many years, and we're already starting to see that happen now. So this is more imperative than ever. As a result of the increase in temperature, the amount of dissolved oxygen is dropping a bit, and that makes things harder for the organisms that are living there. The biggest concern, as more and more carbon dioxide is absorbed by the ocean, and that's one of the ocean's jobs, is to take out a lot of that carbon dioxide and sequester it out of the atmosphere, but it's dealing with so much more now than it ever used to have to deal with. that it's actually changing the pH of the water. pH, a measure of acidity. There's a scale of 1 to 14. 7 is neutral. Ocean water before the Industrial Revolution was about 8.3 on that scale. Then it dropped to about 8.1 just as the Industrial Revolution really started kicking in. Unfortunately, the average water pH along the California coast right now is about 7.75. Now, I will caveat that by saying that California is a unique place in many ways, and one of the things that's actually very unique about the California coast is that the water off California has always been a tiny bit less basic than ocean water almost anywhere else in the world. So the question that we really have to ask is how much less basic can that ocean water go before we start to see negative impact? There are some sites off California coast that have always been extremely less basic. There's a place called Pilgrim Bank just south of here. We refer to it affectionately as Piggy Bank. It has a pH of about 7.4. So it's still that basic side of neutral, but it's close to neutral. And believe it or not, a drop from, say, pH of 8 to a pH of 7 is a 10-fold drop. It's kind of like the Richter scale sort of scenario. It's a logarithmic kind of change. So that kind of a drop is really significant. And to you and I, it doesn't seem like much, but in truth, it actually is rather significant. We also are concerned with, and they're actually taking water samples that will help us deal with and understand a little bit more about what's happening with what we call persistent organic pollutants. These are a lot of the toxic compounds that are washing into our ocean from all sorts of activities that humans are doing. So we call those POPs. We want to know what the levels of POPs are in the water from surface. But what's happening now at great depth is they're starting to show up as well. So this is something we need to pay attention to. There is a little bit of change in seabed topography because of underwater mining, drilling, oil production, all of those sorts of things. We want to know how these organisms are handling these changes now and what potentially could happen in the future. So, let me tell you just a little bit. Although I had originally decided I would talk only about the Gorgonians, I realized that in order to get a really good picture of the whole scenario, you kind of need to know a little bit about sponges too. So, I'm going to tell you a little bit of both. So, what makes a sponge a sponge? Let me just tell you a few interesting things about them. They have no obvious historical connection to any other animal form living on the planet. So we don't really know anything about their origins, who they're related to. They don't seem to be related to anything else. They do not develop any kind of tissue whatsoever. They are massive aggregations of cells, but no actual tissue. They never form a tissue. They are able to produce, however, a protein that's very collagen-like, so the same material that you produce in your skin, they produce, although it's a slightly different chemical process that enables them to do that. They also produce a very interesting protein called spongin fiber, which enables them to actually glom onto and actually pull in very large quantities of water. And they produce these tiny little calcium carbonate or siliceous structures called spicules. And those are the things that will be affected by that change in pH most significantly, because as the water becomes less basic, those little bits of calcium carbonate and silica will degrade in these altered pH states. So we want to know how much can they tolerate before things really get to the point that they can't survive. The other interesting thing about sponges is that they produce a huge array of organic compounds that we don't know the chemical formulas for. We do know that they have a lot of pharmaceutical property, that they actually have the potential to be a whole new array of drugs to be used in the medical industry, but we have to understand a little bit more about their organic makeup first. The biggest thing, perhaps most important thing, is that they pump phenomenal amounts of water through their bodies every single day. And that movement of water is exceedingly important. So while we're talking about them, I'll just point out a couple. This is one right here. This is another one here. We've got some leafy ones over there. This is another one right here. And then the rest of it is the other group that I'm going to talk about. So that brings me to what makes a sea fan or a Gorgonian coral a sea fan. So here we are looking at organisms that produce soft tissue, but in order to maintain the structure that you see, those pretty yellow fan-like things that you see in this particular shot, they have an internal axis, which is relatively stiff. The material is a very strong protein. It looks somewhat comparable to what your fingernails are made out of, but it's a little bit tougher than that, and it's very dense, and it enables them to maintain this shape. They too produce these very small calcium carbonate structures called sclerites that are actually the really only true way to determine one species from another, and that's also true for sponges as well. If you really want to identify a sponge to species, you have to microscopically examine those tiny little bits. Same way for Gorgonian coral. They are rather interesting creatures in that even though you see them in this particular position, if the currents of water flowing down on the ocean floor shift, they will twist their whole body so that they get the maximum amount of their fan facing perpendicular into that current flow. And this is how they feed. They're picking out a lot of suspended materials out of the water, and they will constantly adjust depending on how those currents are flowing, so that they can actually expose a maximum amount of the fan itself to that body of water. We call these modular invertebrates for the simple fact that the sea fan that you see is in this shot is actually composed of hundreds of thousands of tiny little animals. So these are colonial. Each little animal looks like a little tiny sea anemone, but there are literally hundreds of thousands of them that will come together to form just one colony. When I started working with these animals, reading a lot of the older literature, I have to be honest, when I started working with them in the spring of 2002, I didn't really know very much about them. I had worked with pelagic snails and I'd worked with tunicates, but I hadn't worked with gorgonians. The Museum of Natural History had just completed a series of volumes of organisms that were found living on the bottom of the channel. But this group, the Gorgonian group, hadn't been done. It was an unfinished scenario. They hadn't had the time to go back and look at that particular group of organisms. So I went looking for a sabbatical project, and they said, here's a nice little project. It'll, take you about 4 months to probably to, process through our collection and see what we've got. And the estimation at that time was that maybe there were 12 to 15 species living in the region of the California Bight, which is an area of land from Point Conception to the Mexican border. First month in, it was quite evident that 4 months was not going to be long enough to even get a remote handle on this. So here we are, however many years that is later, and I'm still learning a lot about these organisms. But anyway, they are an amazing group of animal, I must say, and very pretty, as you can see. So let me just show you here some pictures to show you some of the characteristics that define the sponges, and then a few characteristics that define the Gorgonians. As I say, we're talking about sponges who are filtering just phenomenal amounts of water. And what they do essentially is purify the water as it's going through the system. As they pump the water in, they're extracting out organic material, but they're actually putting a lot of things back into that water, which then for them might be wastewater, but actually turns out to be enrichment for a lot of other organisms living in close proximity to them. So you'll notice actually in this picture that oftentimes you'll find gorgonians and sponges living in very close association with each other, really close together. They benefit from each other by the filtering processes. But the sponges themselves are just natural water filters. They do a great job at purifying. But they enrich that water, and so when it comes out as wastewater, it becomes food for lots and lots of other organisms. So lots of organisms are dependent upon what those sponges are doing. They shed a lot of their cells very quickly. Those cells settle out on the bottom. Lots of organisms living in the bottom sediments are feeding on those dead cells, so they're getting all of this enriched organic nutrient material that's coming through the sponge's body. So if you're thinking about, a mile down, a mile and a half down, 2 miles down, how much food could there really be drifting down from the surface? Not a whole lot, actually. But of what little is there, sponges are processing a lot of water with a lot of that particulate material in it, and then enrich it, and it becomes a very substantial food source for lots of other organisms, including the Gorgonians themselves. So we're recycling lots of nutrients here, vast recycling of nutrients. In fact, if we think about the best known recyclers, which are bacteria, and we think about all the organic material that's broken down and decomposed by bacteria, sponges are suspected to actually recycle about 10 times more than the bacteria are doing. So sponges are not insignificant creatures living on the bottom. So here are some of those little spicules that I was telling you about. This is the way you identify one particular sponge from another. There are some interesting looking deep water sponges down there. These are a form of glass sponge known as goiter sponges, all those sort of light tan ones. The bright yellow one is a barrel sponge. It might even be a Picasso, what they call a Picasso sponge. I'm not sure why they named it that, but anyway. Here's another shot of them. Little crab hanging out there. He's not stupid. He's going to go where the food is just flowing through constantly, right? The sponges are just pumping water like crazy, moving a lot of food around, and so the animals are going to congregate where that food is. Same way with these guys. This is another kind of sponge. It almost looks like a big flat leaf, but nonetheless it is a sponge, and it's doing exactly the same thing. It's filtering lots and lots and lots of water. These underwater sponges can be actually quite large. One that was collected in a national monument in Hawaii was actually about 12 feet by 7 feet. So that's a big sponge. That's the size of a minivan, if you need any kind of analogy to that. With that extraordinary size, the ability to turn over and manufacture new cells, I mean, new ones are coming in as fast as the older ones are dying. We think that sponges might be able to live literally thousands of years if they're allowed to do what they need to do. So they are indeed what we generate what we call the sponge loop. They're bringing in water, taking things out of it, enriching that water, putting that water back into the environment, and then it becomes food for everybody else. And because they are recycling so much material, this is why we call the sponges the sugar daddies, because they are generating lots of sugar and other organic materials that other organisms can use as food. So the sponge loop is very important. They are places where lots of other organisms like to hang out. So here we have a large brittle star sitting right over the top of the sponge because that's where all the water is being pumped out. Same way here, this sponge looks like it has a hairdo. These are all sea lilies. Here we have another bunch of them. Again, notice they're all hanging around the top. In this one, we have a number of little brittle stars, and there's a few other little things buried down inside the sponge. Same purpose, same rationale. Go where the food is. Here's a goiter sponge with a crab kind of sitting just right in the middle of the whole thing, just taking advantage of all that water and food that's going through. Now the Gorgonians, on the other hand, which is the group I really do work with, create these very three-dimensional type foresty structures on the bottom. And that they grow laterally, they grow in height, they grow front and back. There's a lot of three-dimensionality to them. And that creates a lot of microhabitat for a lot of other organisms that are living down on the deep sea. They too, in addition to those little sclerites, those little calcium carbonate structures that we use to identify them, also produce a very wide array of organic compounds for which, again, we don't know the full chemical formula, but again, those two seem to have a lot of pharmaceutical property associated with them. Now, they're using those compounds primarily to defend themselves and keep things from grazing on them, but there's always exceptions to the rule in nature, and there's always an animal that will figure out a way to get around that. or develop a taste for it, or whatever the case may be. So they are food sources, but that is a means of defense for them. So with this three-dimensional kind of habitat, lots of other organisms tend to congregate around them. These are good places for other organisms to hide temporarily, to shelter, but many of them, we think, are living in them more or less permanently. There's a lot of evidence now coming in that shows that a lot of commercial fish species are using Gorgonian forest as nursery grounds. So this three-dimensionality, the fact that so many other organisms are likely to be congregating around them, this is all sort of anecdotal. Now we want to go out and actually figure out what those relationships are and actually verify and confirm that indeed both the sponges and the Gorgonian corals are really essential for other forms of life living on the deep sea floor. So they do form great trees. This is bubblegum coral, and these trees can be anywhere, well, fully mature, they can be about 300 feet tall, and they are probably about 300 years old. So I wasn't kidding when I said that there are bubblegum trees. These are beautiful, vibrant pink, but very large, very slow growing, but they create phenomenal amounts of three-dimensional habitat for a lot of other organisms. I have to remind you of the fact that these actually are modular invertebrates, to show you a little bit of the actual polyps themselves, these little tiny sea anemone-like creatures that reach out with their tentacles. So as you can see, every single one is out with their tentacles looking for that water current and capturing the food that's traveling in that water. I have a couple of shots here to show you. Here's, if you can see the little shrimp that's hiding in all of this. Just kind of hanging out. Find a good place to hide. Notice the colors blending in nicely, so he's really hard to see. When these animals are collected, oftentimes it's hard to get them to keep the little coral animals themselves extended. So oftentimes when you see museum specimens, you just see these little round pimples where the polyp is actually pulled into the soft tissue that's wrapped around that axis in the middle. So a lot of times, working with material that's already been collected, a lot of times if you don't preserve it quickly in the appropriate kind of material, you lose the extension of the polyps. So that makes things a little, not problematic, but it makes things a little bit harder to identify. So we are going to be very intentional about how we collect samples. We don't necessarily need to collect whole colonies. In fact, we'd rather not do that. I personally only need about 1 inch to 2 inches worth of a branch. That's all I need. I can get more than enough sclerites out of that to identify it to a species, and that way we haven't significantly impacted the forests that they are a part of. So here's another shot of the bubblegum coral, where all you see are these kind of funny little pimples where the polyps would have extended out if they had been preserved a little bit differently. I actually work on, right now, my focus is on a genus that everybody has sort of, it's too complicated, we'll put it on the back burner and deal with it later. And I sort of just got thrown into it. There was no information, I didn't have anything to work with. So I thought, well, in order to understand what's going on in terms of what we have in the collection here in Santa Barbara, I'm going to have to tackle this particular genus, which is the genus Swiftia, which I think has something to do with the fact that it's so amenable to shifting itself to catch those water currents. Swiftia means flowing or swift. So anyway, so some of them are very extensively branched like this. As you can see, all the little polyps are extended, but then you have some that look like this. And lots of people were putting that in an entirely different genus than this one. But if you look at the sclerites, you come to discover that, oh yeah, okay, it's in the same genus, but maybe not the same species. So I have been working on this group for a very long time. And as I say, it's a genus that everybody has pretty much just wanted to sort of ignore and hope that it went away, but it didn't. And it's actually quite abundant at depth, so we can't really ignore it anymore. So I am starting to work on this group fairly intentionally. So whenever anyone goes out collecting, whether it's NOAA or the Ocean Exploration Trust, which runs the Nautilus Live program, or any of those other deepwater expeditions, they all know that if they find anything that's red, they need to collect something from it, because I need to know what's going on. These are the little sclerites, actually, that are found in these two species, as you can see. So they have some interesting looking characteristic sclerites. The ones that look like big bacterial rods are actually very, very tiny, but those are characteristic of the genus. And so that's how I know it's in that particular genus. And then you have to look at a little more subtle detail to figure out which species is which, but nonetheless. This is another one that has not been reported for Santa Barbara or anywhere along the California coast until about 5 years ago. A colleague out at UCSB by the name of Milton Love, who does a lot of fish surveys out there. They get a lot of Gorgonian as bycatch, and he contacted me and said, look, if we get any of that, do you want it? And I said, you bet I do. So this bright gold one apparently is quite abundant at depth, and you see more and more of it as we go deeper and deeper and we look at more areas that we haven't surveyed before. And it is really this vibrant gold color. But again, notice there's several little fish hanging around, and so we're interested in what's going on there in terms of interaction. These are the sclerites that come out of that bright gold colored Gorgonian. These are interesting. They're maybe 5 millimeters long. That's big for a Gorgonian sclerite. And I made the mistake the first time of handling specimens that Milton had collected without wearing any gloves. And that was a big mistake, because a lot of these little sclerites got under the skin between my nail and my finger. It was like handling fiberglass. Yeah. So I learned the hard way. Got to wear gloves on that particular one. But they have these very long sclerites, and they do make what almost looks like a little crown of spines around the top of each polyp. You can sort of make that out. Most of this is actually tentacle, but there is a crown, literally, of spines that go all the way around the top of the colony, or around the top of each polyp in the colony. And yeah, so that one was reported from Japan, but apparently is more abundant on this side of the Pacific than we thought. And you do get some of that cross transoceanic distribution. Kind of interesting to think about how that might have happened, but that's another question for another time. Here's a third deepwater form. This is, it's basically sort of a creamy white color. Again, it attracts a lot of other organisms. The two that you see attached here, these are funky, almost parasitic sea anemones that you see attached to them. And their sclerites kind of look like this. They form more like a scale type structure. And then all of these little bits are embedded in the soft tissue of these animals. And I think I have one, yeah, this is another one that has not been recorded previous to my working with them. This is another genus that has shown up at deeper depth, Placogorgia, and it's kind of this fuzzy pink looking thing. Here's another shot. This one was actually a specimen that Milton did collect for me when he was out doing his fish surveys. And they have even stranger looking sclerites. You've got these sort of weird looking things. Yeah, so anyway. So animals that are living in association with Gorgonians turn out to be a lot of species of rockfish. So many of you, I think, are aware of the fact that one of the reasons why marine protected areas here in Santa Barbara County and the sanctuary, the marine sanctuary, were established was to help ensure that populations of rockfish had a chance to recover from overharvesting. And those rockfish species are still, they got a long way to go, but they're doing very, very well. When the marine protected areas came into being, it was estimated it would take about 5 years for those populations to become large enough to start spilling over out of the boundary of the marine protected area and then start populating surrounding water, which fishermen would then have access to. They can't fish in the marine protected area, but they can certainly fish in areas adjacent to them. They thought it would be about 5 years before we saw a lot of spillover. It only took like a year and a half. So there's a great deal of resiliency out there if you give nature a chance to do it. Here are some brittle stars attached to Gorgonian coral. Here are sea lilies and crinoids attached to a number of them. Here's one of those big bubblegum trees with a lot of brittle stars attached to them. Here's a squat lobster. These are fun. They're kind of cool looking. Nudibranchs. Yeah, here's one eating one of my specimens. But anyway, as I say, even though they have the sclerites and they have the organic compounds, there are still animals that can kind of override that. But it turns out to be a very species-specific sort of scenario. So there's only certain nudibranchs that will feed on a certain species of sponge or a certain species of Gorgonian and nothing else. So there's no competition for those resources. But yes, this guy is stripping off my soft tissue. Then you have, obviously, a lot more interesting creatures, like this little Deepwater octopus that was just kind of hanging around the edges of some of these Gorgonian colonies. And you even have, at that depth, you actually have larger vertebrates like cat shark and things like that who are actually tethering their eggs to those Gorgonian corals. It's a lot safer for the developing embryos in those little egg cases to be kind of hidden in amongst the forest, as it were, and gives them a chance to develop to the point where they can hatch out and become adults. So this is a lot of cat shark egg cases laid on this particular Gorgonian. Now, I have to ask you this question. What seems kind of odd or unexpected about the organisms I just showed you? Color. Who would have thought that a mile and a half or more down, where there's absolutely no light at all, except for a little glow coming from some volcanic material, that you would have bubblegum pink and bright yellow and orange and purple and yeah. So when the first expeditions were done and people turned on the lights, it was like, oh my gosh, what is going on? Because nobody ever sees those colors. I mean, if you're down there at depth with the lights turned off, which is what all the other animals are seeing, you don't see any of the color. So that was a big surprise, a delightful surprise, but a big one. The other thing that's a little odd is the fact that some of these things are growing so incredibly large in an environment that's typically known as not being a hugely rich, abundant nutritional pool. So yeah, you kind of scratch your head and go, gosh, just how much food is there down there? Because we're looking at some really, really large organisms. So those two things struck me right off the bat, was that I never would have expected to see that kind of color. And they retain their color even after they're collected, which makes them even more special. Yeah, it's beautiful. It's wonderful to just walk into the museum and, if it's been a really rough day, pull out a tray and you just see all these pretty colors. It's amazing. And it just goes to show that for all that we know, there's so much more that we don't know. And if I have learned nothing else in working with these organisms for the years that I've been working with them, it's that. Mark was overly generous in saying that I'm an expert. I feel so not an expert on these guys because there's so much more I need to learn. There's so much we don't know. But it is interesting to listen to a lot of the NOAA staff talk. There was I'm going to show you another interesting one here. We had a 2-day workshop out at UCSB in April, and that was the first meeting that we all had together to determine how are we going to set priorities for where we're going to do these surveys. I mean, we've got hundreds of thousands of miles worth of bottom out there, but it's a 2-year project, and the money ain't going to go that far. So we have to prioritize where we're going to look. And the one thing that kept coming up in the conversation was, Beth, what are you going to do when you retire? Because aside from myself, there's a gentleman that works at the California Academy of Sciences up in San Francisco. He's older than I am, and he's probably going to be retiring in the next few years. Then there's me, and then there's really no one else working with Gorgonians on this coast until you get down to Costa Rica, and they typically tend to work with species that are found in Central America. So, the bottom line was they're experts at running the equipment, and they're experts at navigating the bottom, mapping the bottom, understanding the physical features of the bottom and how water would flow and all of those sorts of things. But they don't necessarily know everything that they're looking at. They're not trained that way. So they kept sort of reminding me of the fact that when they start collecting, guess who's going to get it? So we'll have to talk about my my time allotment. I'm going to be probably overwhelmed with the amount of stuff that they're actually going to start sending to me over the course of this 2-year program, which is fine. I mean, I'm happy that we're getting material that will answer some questions. There's some really strange things, as I say, going on where Point Conception is, because that's kind of the transition point between southern forms and northern forms, and there's some really weird stuff going on there. But yeah, it takes a lot of time, and it's doing this kind of work is not my full-time job. You know, it doesn't pay the rent. So I got to figure out how to allocate my time a little more wisely. But it is fun to do. It's like piecing together a puzzle with a million pieces, and you're trying to figure out how all this fits together. You have some information that's coming through old literature, but the old literature is usually not in English. It's in German or it's in French. Some of it's in Spanish. A lot of it's in Japanese. No, I don't read Japanese. And they were working with very small parts of one colony, trying to understand more about these organisms. And they didn't have the technology that we have today to actually go 1.5 miles down or deeper and actually see them in sight and see what they're doing and how they're responding to the other organisms around them. So the bottom line was we need the taxonomists, which is what I am, and they said, you can't retire, and you can't retire because you're never going to run out of things to do, which is really true. We are finding a lot of cool things. This is a bamboo coral. It is a type of Gorgonian. We've never seen one sort of that looks kind of like this. It's kind of like a lyre, you know, if you think about the musical instrument. It's kind of a weird-looking bamboo coral. It doesn't look like bamboo corals we normally see. This one's interesting. Anybody want to tell me what that is? Got any idea? Yeah, it kind of looks like a nudibranch. That's a good guess. No cigar, but it's yeah. It's actually a sponge. It's called golf ball sponge. That doesn't look like any sponge I've ever seen, but there are lots of things down there that don't look like anything anybody's ever seen. How about that? It does look like a Venus flytrap, and actually Venus flytrap is in the name. It's neither a Gorgonian or a sponge, but I thought I'd throw it in there just for good measure because there's so many weird things down there. It's actually a sea anemone. It's called the Venus flytrap anemone. Yeah, so it will be really exciting to see NOAA plying the waters up and down the coast over the next 2 years. Lots of talk about how they actually managed to get the money, which I'm not privy to all of the ins and outs of how that happened, but anyway. But my role in all of this is basically to examine all the samples that are being collected of Gorgonian coral. I'm emphasizing the red ones, but they'll give me everything that they are able to sample and identify them. So between me and Gary up in San Francisco, we will have lots of work to do. Are we likely to encounter new species? Oh yeah, for sure, yeah. That's kind of the fun part of the whole thing, is realizing that if you've looked at it a bazillion times and compared it to so many other things, and you go, it doesn't look like anything else, it has to be new. That's pretty exciting. Yeah, you get to name it, you know, that's kind of fun. But there's a lot there. I remember my father and I used to have a lot of conversations about how money is spent. My dad was in the aerospace industry, so you can kind of figure where his leanings were. But I told him, I said, Dad, I understand the fascination with space and all of those things, but I don't think many of us are going to ever be able to live there. I think we need to pull that money back and pay attention to the planet that really does support us. And these organisms are a vital part of ensuring that life persists on this planet. And if we don't understand how they do that job, and we don't understand how they're being affected by the things that we do that are harming them, then we're kind of shooting ourselves in the foot. And this work is really important. Sorry, get a little emotional, but this is my passion. This is what I do. And I love the creation. And I love the marine aspect of it so much that this work is so important. So as you're walking along the beach, think about all the organisms that you can't see. And in actuality, that's one of the biggest problems, is that you can go to the beach every single day, and yeah, it'll look a little different depending on the cloud cover, the season of the year, that sort of thing, but it still looks like the ocean. There's no hint to what's going on down below the surface. It's sort of out of sight, and because it's out of sight, it's out of mind. And people really don't often think about what is actually going on down there. And for good or bad, what goes on down there is what enables us to live terrestrially. So that's kind of the main motivation for me ruining my eyesight, looking at all of these. Anyway, so I want to give credit where credit is due. NOAA is phenomenal when it comes to doing both still and video. They actually post a lot of their images online. They have a whole archival system so that you can see what they're seeing. And there's, as Mark mentioned, I do a lot of consulting work for a lot of the different satellite NOAA offices throughout the country, on both coasts, actually, as it turns out. So these are all sources of images that I was able to pull together. I do want to say that were it not for my research students, I would have a much harder time getting any of this work done. One of them is sitting in the audience tonight. But I've had some really phenomenal research students, one of whom graduated about 2 years ago, and I was so sad when he graduated because he was so good at doing what I needed him to do. And I tried to convince him that he didn't need to go into medicine, but he decided that's what he was going to do. But he really had a gift that was rare. But I think, thinking back over the 5 or 6 students that I've had working with me on this project since 2002, all of them have very quickly developed a really keen eye for what they need to be able to look at and see and be able to discriminate one from another, which is essential. And so I'm really grateful for those students and their work. So, yes?
I understand that in Hawaii, certain sunscreens
Yes.
have been banned because of their effects on the coral life there. Do you have any concerns?
Yeah, I think it's not, at least, at the point where we're at right now, I don't think that's going to be a huge problem at the depth that we're talking about. But, all that stuff does kind of percolate down through the system, and a lot of those compounds don't degrade very easily. So, in time, those things will work their way down, and yeah, it is a little bit concerning. I mean, anytime you're putting any kind of chemical in a body of water, water does all kinds of interesting things. It dissolves them, it reconfigures them, there's all sorts of interesting chemical reactions that take place. So, yeah, it's a problem. I mean, I think in time it's going to reveal itself more so in shallow water, in the initial stages. I mean, we're already sort of seeing the impact of some of that stuff on stony corals, the ones that build the huge reef systems in the shallow waters and tropical areas. It's already had a negative impact. So, what do? You got to find something to keep yourself from getting skin cancer, but on the other hand, those things do add negative solutions to the ocean where they don't belong. So, yeah. Yes, sir.
You said something that sounded like sclerites are the only way that you can distinguish one species from another. Is that why sclerites is that why they rule when it comes to your taxonomy?
Mm-hmm. I mean, yeah. And in fact, if you go back in the old literature, a lot of people were sort of, if I can go back, here, let me do this. Let me show you one thing here. If you go back into the old literature, a lot of invertebrates that you find, if you just go tide pooling, and you know what you're looking at, you can probably identify most everything that's out there, and you don't really need to look at them microscopically. Sponges, no. Gorgonians, no. Because a lot of times you'll have 2 or 3 specimens laying in front of you. They all look morphologically the same. When you look at the whole colony, they're the same color, they got the same branching pattern. But you look at the sclerites and they're entirely different genera. And I learned the hard way that you can't eyeball these guys at all, which is why I've sort of trained, once I trained them, but they have a better idea now. I always tell the NOAA folk, look, if you're gonna go out, it's fine that you take still images, it's fine that you take video, but I can't tell you definitively what species it is unless I see the sclerites. Yeah.
Just to follow up, is there any way of distinguishing varieties within a species?
Yes, there is. How do I explain this in a way that doesn't sound sort of nebulous? When you've looked at thousands of specimens, after a while, you just kind of know. You do. I don't know how else to explain this to you. You do, after a while, you sort of just sort of develop this inner sense that says, no, these are not like these, and and that's how it's done.
I hate to see you apologize for that because it sounds like something analogous to facial recognition.
Yeah. No, and to be perfectly honest with you, when I first started working with these, I didn't have any sense of this at all. And one of my colleagues from the Smithsonian came out because he was so excited that somebody actually was on this coast was interested in working with these animals. And I showed him the collection, and he said, well, first, obviously, you just how are you doing this without enough specimens? You just don't have enough material to work with. He said, this is like you're working in a vacuum. And I said, oh, am I? What do I know? And then he said, are you able to distinguish, the subtle variations? And I said, well, no, not at this moment. So here's what I do to kind of help you understand what I do. When I have, say, 100 specimens, we will take sclerites from every single one of those colonies. We'll photograph them. And then we will lay out all the photographs on a table. I know this sounds very old-fashioned and quaint, but this is the best way to do it because you can look at all the sclerites at the same time. They're all of the same resolution, they're all the same scale. And you start kind of lumping them, okay, these sclerites and these sclerites here, they look really the same. Then you make another pile here and another pile here. Then you go back, look at the literature, look at images that others have done elsewhere, either in time or at some other institution, and then you begin to sort of just get the rhythm for the variations in these things. Yeah, and I say, like I say, it's some sort of, it's kind of nebulous, but But for a long time, I didn't know that, and I was really frustrated by the fact that I couldn't sense these variations at all. But all of a sudden, it was like, oh, okay, I get this now. And I could start to see those variations a lot easier. So I think it's just something that comes with looking at lots and lots and lots and lots. And there were some specimens in the museum collection here. I probably did sclerite samples on them at least a dozen times. And every time I looked at the sclerites, no, it's not that. No, it's going to be this. And then I'd set it away, I'd put it on the side for a couple of months, and I'd come back and look at it again, and I go, oh, no, it's not that either. And you just keep doing it, and you keep doing it, and you keep doing it, and you keep doing it, until finally, the little light bulb finally goes on. But, yeah.
Now,
Yes?
every picture I looked at, I wondered what the depth, how deep it was.
They didn't give me all of that information, but they tend to work anywhere between 500 meters, which is about, 1,500 feet, 1,600 feet, to anything below that. Yeah, but I don't have, for a lot of these images, I don't know the exact depth. Sometimes they don't post all that information. When I get the material and they send me all the provenance for everything that's collected, then I know what the depths are. Yeah, they're trying to work within about 500 meters to about 1,500 meters.
About the coral. What's happening with the Great Coral Reef?
The Great Barrier Reef, you mean, in Australia? Yeah. Well, about a third of it's dead entirely, and I'd say another good third to maybe even a little more than that is really hurting. Yeah.
It's dying.
Yeah, it's dying. And it's basically the result of increases in water temperature more than anything else. So now, I mean, that's very concerning, considering how many animals are supported by that reef. But when you start seeing those kinds of changes taking place at depth, where we thought, oh, the deep ocean, it'll survive this, it'll essentially be insulated from all of these things that are going on at the surface. No, that's not the case. So, yeah. Yes.
Yeah, such an important subject that you're dealing with, and very, very interesting. I want to thank you for
Oh, thank you.
and a beautiful presentation.
Thank you.
It seems like to identify the Gorgonians, you're using morphology of these sclerites. So I have two little questions.
Okay.
One was, can you tell us a little bit about the microscopy that you're using?
Okay.
And then the other thing is, I was wondering about the actual chemical makeup, if you ever look at that.
Yes. The second one's easy to answer. It's mostly calcium carbonate, a little bit of aragonite thrown in for good measure, but it's primarily a calcified matrix kind of material, so easily degradable if it if it encounters drops in pH, that sort of thing. And
They don't differ from each other.
No, they really don't. The axis will a bit in terms of the construction of the axis that supports the entire colony. That will have varying degrees of different types of compounds, but the sclerites themselves are pretty much uniform in terms of their makeup. The sclerites, I will say, vary depending on the quality of the water that the colony has been living in. Under certain circumstances, they might make their sclerites overall thicker, and in other circumstances, the sclerites will be thinner. That makes for confusion right there, because you have to sort of accommodate or make adjustments for the fact that you're looking at environmental factors that are dictating how the sclerites are formed. So that gets confusing. And I didn't know that either when I started looking at them. I thought, well, why are these really, really fat and these are really, really thin? Not recognizing the fact that there's a huge amount of environmental influence on the production of the sclerites. Now, to make these, let me show you one here. To make these sclerite preps, you take a little piece of the branch and a razor blade, you just slice off a tiny bit of the soft tissue away from the axis. I use these deep well slides. They have a big bowl in them, basically. Put them in there, drop about 15 drops of bleach. Plain old bleach, household bleach. Nothing fancy. And you wait for the soft tissue to dissolve, which it does pretty readily in the bleach. And what you have left laying on the bottom of the well are all these little sclerites. You can see these little tiny little bits. Flush them with water, maybe 2 or 3 times to make sure you get all the bleach off of them. Flush them 2 or 3 times with 70% ethanol. And then the last flush, you use that ethanol, you just suck all the sclerites up in the ethanol and drop them on a microscope slide. Let them air dry. I don't put cover slips on them. Lots of people do, but I don't put cover slips on my sclerite preps because putting the cover slip on crushes the sclerites and you don't get a true look at what the sclerites are really supposed to look like. You're getting lots of little broken bits. I need to see the sclerites whole. So that was kind of my contribution to the whole sclerite prep deal. I kept telling people, don't put the cover slips on them, don't put the cover slips on them, because you're breaking a lot of the sclerites, and then we're not seeing the definitive features. So it's not high tech. It's very, it's real basic. A little bleach goes a long way, but that's how I prepare them. Now, if I'm going to do SEM, and I didn't bring any of my SEM images, but if I'm going to do SEM, I do an additional alcohol wash, and then I do one of 98% ethanol, which is what my samples go in if we're going to do any kind of molecular sequencing. So when it's collected, if I tell them, no, I only need it for morphology, but if I need it for DNA analysis, which I send up to my colleague up in Washington, it needs to go into 98% from the get-go. But when I get ready to put them on the little stub, I wash them with 98% ethanol, and then I do a water wash because the ethanol has a tendency to eat the adhesive on the stub, and they get embedded in the goo, and then you can't see them well. So I also learned that the hard way. But when I'm just ready to actually drop the whole suspension of sclerites on the SEM stub, it's just pure water. It's purified water, and then they stick to the surface, but they don't sink into the adhesive because the alcohol's not dissolving it. And that's how I do it. Yeah.
Do you have a scanning electron microscope?
The Museum of Natural History does. Yeah. It's very small. It's not like I remember when I was in graduate school, scanning electron microscopes took a huge room to house it all. This is a tiny little thing. It looks like about the size of a washing machine with some few other things attached to it. But basically, it's a very small machine, but it does phenomenal work. I don't actually I do all the prep work on the stubs and get them ready. Daniel Geiger is kind of the SEM guy. He's really good at getting really nice images. No matter how lousy your mount happens to be, he can get some really nice stuff off the stub. Thank God for Daniel. But it's a really nice little machine. And when the first time I saw it, I thought, no, this can't be. But yeah, they've come a long way in terms of building those things. So the museum has one. People from UCSB come to the museum to use it because I don't think there's another one in town. Okay.
How big is that? I can't see.
That is a 50 micrometer scale bar.
So how long would that be? How many micrometers do you need to make, a centimeter?
Oh gosh, now you're asking me to do my math, which I'm never very good at. I'd have to sit down. I'd have to punch it out on. These are pretty tiny, yeah. You can see them with the naked eye, but you can't see any of the sculptural detail or anything like that unless But yeah, very rarely do you get one that's even a millimeter, and you get really excited when you get one that big. Yes, ma'am.
I wanted to add, since there's some interest about the electronic microscope, that one day a year, the Museum of Natural History has a Science on Site Day where they feature that machine, and you can come into the museum, sign up, and we'll be glad to walk you back over there, and Daniel will sit you in a little circle, explain the whole damn thing, answer every question that you might have, and then, and you can sign up and go through it again. So watch for that on the museum's website, on the Science on Site, that says the microscope.
Yeah, it's a great little machine. Yes, ma'am.
You initially said that they were estimating 12 to 15 species.
Yes.
How many are you up to now?
Okay. Spread over about 23 genera, right within the bight itself, which is only from Point Conception to the Mexican border and only out to the backside of the Channel Islands. I think I'm at like 45. Yeah, and I think that I'm missing a lot. And that's just based on material that's in museums. Okay, there's a whole lot of stuff that's out there that probably hasn't been collected yet. So who knows how many. But the California bight in and of itself is a very biologically rich and diverse area in every other animal group. So there's no reason to think that the Gorgonians wouldn't be as well. But yeah, that was when I was told, well, it's 12, 15 species. It won't take you too long. Yeah. Well, yeah, there are a lot more than that. And that's, as I say, that's just working with material in the Santa Barbara Museum of Natural History. I've been to the Smithsonian several times. I have no idea how much more they have because a lot of their material, they constantly collect, but the Smithsonian has so few curators now because that when they retire, they're not replaced. The government won't pay for their curation anymore, so that once a guy retires, you don't see any replacement for it. I mean, I could spend months just going through cabinets of stuff that's not been identified. And who knows what's in there. I mean, that's where actually I found the first species that I described as a new species. We had one piece of it here in our museum. Nobody could tell me what it was. And I'm like, crap, what do I do now? I only have one specimen. I have nothing to compare it against. It doesn't look like anything else I've seen. The second time I went out to the Smithsonian in that period of time, Stephen, who is one of the curators, he said, well, if you have any time left over before you leave at the end of the week, can you look through some of the cabinets back there that, we got all the stuff back there and we don't know what anything is. So I said, yeah, sure, fine. So I did. I took half an afternoon and I kind of was rummaging through the drawers, and you probably could have heard me clear down to Florida when I found 2 more large samples of the stuff that we had in our collection that nobody seemed to know what it was. So how many more things are hidden in drawers in the Smithsonian? A lot. I'm quite convinced of that. But there's no bodies to process it. So yeah, who knows what else is there. But yeah, it's about 40 to 45. And after this 2-year run, who knows how many more I'll get in that. Yeah. Yes,
I was wondering, do you have any hypothesis about the crazy colors, why they're so colorful?
I don't. And I was just talking to someone about that the other day, because it makes absolutely no sense. I mean, it takes energy to make all that pigment, But wow. I mean, it's just amazing. I mean, there are very few organisms that generate at that depth. I mean, if you talk about mid-column depth, maybe 500 feet to maybe 1,000 feet, you'll get organisms that produce bioluminescent light, but that's not real bright, intense light, and that's pelagic. That's not down at the bottom. I have no real good scientific explanation for the color, but hey, something may yet turn up, but I don't know. There's no means to see it. The wavelengths that would be generated down at that depth would not capture all that light. And I mean, the first time you turn on those lights on the ROV, it's like, it's like, what's gonna? So, no, I don't. I'm thinking about it a lot though, because a lot of my students comment on the fact that, gosh, it's so dark down there. How does anybody see that? And I said, because nobody does see that. Unless we go down with our technology, and suddenly it's...
Must surprise the fish when you come down there with your lights.
Yeah.
Well, that's the other thing. You know, a lot of deep sea organisms have very reduced eyes because there's no light to use them with anyway. But you do find a fair number of species of fish with very well developed eyes, and you turn on the lights and they're all like, whoa! They're all like right there. You know, like, wow, this is really cool. You know, never seen any of this before. But because, you know, what are they using to use the eye? And it's a very well developed eye. It's got all the requisite parts you need to see, but there's no light to use them with. So I don't know. You know, these are the things that are on my long list of things to ask my maker when I hopefully get there. And, you know, okay, now why this, you know, and how come this? And the list is getting longer all the time. But yeah, your guess is as good as mine. I really don't know, but it certainly makes my work a lot more fun. Yes, sir.
Well, you mentioned if anything in the source of life would be glowing from magma. Looking at the deep sea floor, obviously there's heat, there's going to be change in everything. Is there a sudden change in the fauna, the flora?
Yes, and I should qualify by saying that, you know, where those areas are where you have very weak spots in the Earth's crust and you do see some of that magma action coming up, the water right around that, these are these hydrothermal vents that you've probably heard about, you know, the water there, it's hot enough to melt any kind of heat probe you've got. But you go just a few feet away from that, it's like 75 degrees. It's like bathtub water. Then you go just a little bit further and it's dang cold. Yeah. So it's very patchy, you know, and huge expanses of the ocean floor are just flat plains with, you know, these big seamounts, but no other means of temperature generation, just those little isolated patches. But I was talking to a student just the other day. We were talking about, you know, how much of the deep sea floor have we actually, you know, explored. In total on the planet, about 4%. So when my NOAA friends were not kidding when they said, "Oh, you're going to have plenty to do for the rest of your life." So we only looked at like 4% at the bottom, and you know, they have to be so intentional about where they choose to do the surveys in this 2-year run because you can't do it all, and the money's only going to go so far. So we're trying to help them figure out where's the best bang for the buck.
Yeah, well, and obviously you got to hone that down, but you know, I'm just thinking, what about core samples in these flat areas?
Well, they're doing a lot of they're doing core sampling, they're doing water sampling.
Correlate.
Yeah, we're trying to see if there's any kind of correlations between the organisms that you're seeing, the temperature, the topography, the sediment, the dissolved particulates in the water. Is any of this coming together? Are we seeing any kind of patterns? Exactly. Going to take a long time to accumulate that much data to actually get the picture. Yeah. I'll be long dead before that happens. Okay. Any others? Well, let me just do my last little. Thank you for coming tonight.